All right, here we go. So once again, I did this video, and the microarray experiment is longer than our other videos, but I decided to break it up. But I did this video an hour and 15 minutes, and there was no audio. So frustratingly, I got a practice run, I guess. But I can see my audio now. It's recording. And after going through it, I found a way to break it into 3 videos. So all that being said, here we go. We're going to start. You can add this to your bioinformatics handbook, but things are getting a little cluttered for me to kind of show you. So I'm actually starting a new R Markdown file, but I'd like you to add it to your previous R Markdown file. So the first thing we're going to do is we're going to install the required packages. And so the first thing we want to do, and I'm going to add these packages for you to just copy and paste, because it's a lot of typing and it can be a lot of opportunities for typos. So what I'm going to do is I'm going to put it down in the description below, and you can just copy and paste them in. But so these first sets of packages up here, gplots we're going to use in the third video, the last video. This is a way that we can make a nice heat map of our data. Don't worry about it right now. BiocManager is another installation sub-package. So if we go up here and we install packages, you can see that the repository is crayon, and that's where a lot of like the very general packages, plier, ggplot, and all these things that are used by a lot of people, stats packages and stuff. But for biology, we have our own kind of separate repository where there's a lot of biology specific packages, and that's called BioConductor. So BiocManager is what we use to access BioConductor and get the packages that we need. So now that we have BioConductor, and what I'm going to do here, since I already have them all installed, I'm going to put eval equals false. So after you install them the first time, you don't have to run the installations again, because it takes some time. And I don't want you to sit here and just watch packages install. So these are all the packages we need to install for this microarray experiment. These 2 ATH ones are kind of database files for Arabidopsis, and that's the plant we're going to use for this gene expression study. And in here they have kind of descriptions of what the gene is, the gene symbol, the TAIR number, which is the Arabidopsis information network. It also has another identification number. I don't remember if it's NTRES or whatnot. So each database always has their own numbers. So we have to like match them up, right? And so and the same thing here with this org.At.tair database. This gives us more information of the genes that we're going to identify. Oligo, affy, and affy, these are packages related to analyzing the microarray data. Limma, I've heard it pronounced both ways, is how we're going to use kind of this linear regression to determine the differential expression between our 2 data sets. Stats, obviously statistics. Reshape is a way that a package that allows us to manipulate a data frame. And so those are kind of the main players that we're going to use. So now let me check my notes, make sure I've explained everything. Okay, so the next thing that we have to do is we're going to have to load these, right? Because we've installed them, but they're not in our R instance currently. So what we're going to do is we're going to take this, copy it, if it'll let me copy it. Whoa, didn't want to do that. All right, we're going to copy it, and then we're going to paste into the next section. And I'm going to show you a handy little tool here. So what we want to do is go click on the magnifying glass up here, and we're going to find BiocC BiocManager, and then ::install. And we're going to make sure that you have selected here in section, because we don't want to find them all. We just want to find them in this code chunk that we have highlighted. And then we're going to replace with library and hit all. And it says we've done 10 occurrences. Perfect. So now we've added the opening library for all of them. I'm also going to add to the top here the gplots, because I forgot that one. Okay, so I'm gonna hit this. Is it gonna run? Oh, I gotta go to my console. I'm on R Markdown. Okay, so let's get rid. Oh crap. All right, let's see if I can undo whatever I just did. Man, this is off to a hot start, huh? All right, here we go. Let's hit run, and we're gonna load all of our libraries here, and we are set to do analysis. Now, what are we gonna do analysis on? So let's go to the Googles. We're gonna type in Genelab. So what Genelab is a data repository from NASA for things pertaining to NASA, like spaceflight experiments. And that's kind of my expertise, is plant space biology. So Genelab is this really nice curated data repository. And we're going to go to data repository under data and tools. And we are going to search for BRIC, BRIC 16, and hit go. And so BRIC, as you can see here, stands for Biological Research in Canisters. And so the way this works is NASA has these big cylinders, these canisters, that hold Petri dishes, right? The regular 90 millimeter Petri dishes. And for these experiments, what you do is you put your Petri dishes into the canister and seal them up. And then SpaceX sends a rocket to the space station. And on that rocket is the BRIC. The astronauts take it off, and then they just set it aside. It's nice because there's no need for the astronauts to actually do some of the experiments. Crew time's a big limited resource for spaceflight experiments. And then at the end of the experiment, you can freeze them and put them at -80 degrees to preserve all the RNA that is present in the sample, so the gene expression, our measure of gene expression, or BRIC has this thing called the PDFU, which is the Petri dish fixation unit, which allows you to put a preservative in there. In this case, there's preservative is called RNA later, which actually kind of freezes all the RNA and preserves it so we can measure it after it gets sent back down to Earth. So this experiment that we're doing was conducted by a colleague of mine, John Kiss, who's at UNC Greensboro. And John's been doing space experiments since the early 90s, so before most of you were born, likely. And so we're going to click here, but make sure, so there's multiple BRIC experiments, especially BRIC 16 experiments. Make sure you click on the one where the PI is John Kiss, because there's one for Sarah Wyatt, who's at Ohio University, and there's also one, I believe, for Simon Gilroy. This one is from Alison Blankenfor. But I believe there's one for Simon Gilroy at Wisconsin too. So we're just going to click on this one. And so the nice thing, Genelab does a very, very good job of annotating the data. And so this is all publicly available data. And as you can see here, the factors that the study looks at is how plants grow in space compared to ground, and their exposure to cosmic radiation as well as spaceflight. It's a DNA microarray experiment, and it uses affy metrics. That's that affy package. Affymetrix is a company, probably the most prominent company that makes microarray chips, but there are others too. And depending on the platform or the chip that you use, you would analyze it differently. So it's important to look for this Affymetrix chip for this. The grant he was funded on. So this was actually flown on STS-131. So STS is the signature for spaceflight experiments. Spaceflight experiments, for shuttle missions. And so this was actually flown back before the shuttle was decommissioned. And so here we have all this information on this study. So we have how the samples were collected, how the RNA was extracted, how the labeling was done for the fluorescence, how the hybridization was done, the data collection, etc. Everything to ensure that this is the right data set that you're looking for. Okay, so now we come down here to samples. And as you can see, we have 6 different samples. We have 3 that are flight, and we have 3 that are ground. And if we go over actually, we're going to go down to this next one here. If you go over, we have this array data file, this array design. So these are showing what the actual the experimenter or PI of the experiment did. But what we're interested in is this array data file. And so as you can see, this is the raw cell file. And what that means is this is the data that was taken off of the microarray. So it hasn't been processed. But Genelab, they have their own consensus pipeline that's been built. And they actually go through and process this data, and you can actually download the data after it's already been processed. But what's the point in that? Because we need to learn it, right? So what you're going to do is you're going to click on this. And one thing I want to point out, it's very important that you download to the same folder that your R Markdown file is in. And if you don't know where your R Markdown file is, we're going to get rid of this. You can go to Session, and you can say Set Working Directory to Source File Location. And what that does not have an active source file. Oh, I have to save this first. All right, so I'm going to save it. I'm actually going to save it where I know my other stuff is. Here's my NASA folder. NASA, meta-analysis, microarray, brick 16. Here we go. And I'm going to call this microarray brick 16. Okay. All right, so now if I go to Session and I go to Set Working Directory to Source File Location, it's going to tell me where I have my R Markdown file. And so you need to save your data to the same file or the same location. Alternatively, you could save it somewhere else and then just include the full file path when you load data, but it's easier just to save it to the same location. So I'm going to go back and I'm going to click on this raw.cell.gz, and what I want to do is I'm going to actually change the name of this because this is a very cumbersome long name. I'm actually going to change this to FL1. So this is the first flight, right? So we have FLT means flight. So I'm just going to say FL1.cell. And then for rep 2, I'm going to call it FL2.cell, FL3.cell, and then ground, I'm going to say GR1, GR2, and GR3. I'm not going to download them all because I already have, but I want to show you what this ends up will end up looking like. So if I go, I'll bring it over here in a second. So once you download those files, they are .gz files. So they are, they're zipped. You need to unzip them because they're packaged. You can't open them without that. So once you unzip them, eventually what you should have is 3 files, F1, F2, and F3, or FL1, FL2, FL3, and then G1, G2, G3. So F1, F2, F3, G1, G2, G3. And make sure that you label the ground controls as ground controls and the flight as flight. And don't mix that up. So this will save us some headache later by naming them like that. Okay, and with that, we are done with part 1. So we have all of our stuff installed and we downloaded our files. And up next, we will start annotating and normalizing our data.